Okay.
On a talk, it's a very emerging topic, new concepts that's going to change the way we are practicing agriculture in the future. Yours Raimaker is a well-known distinguished professor in the field of microbiology and more so in microbiomes. And he is a director of microbial ecology. He stands for Netherlands Institute of Ecology, and he is also a professor in Leiden University. And this program is going on for the last 8 days. They landed here on 18th of this month, and we had 1 week of intense program. Started on 20th officially, and it's going on, and it is going on until the evening. So, the presence of those in IRI, I would like to extend the benefit to the rest of the students and faculty members who is not a part of this program. My responsibility is just to introduce the speaker here. Those who are working on microbiomes and microbiology, soil microbiology, rhizosphere microbiology, Pius Raimaker is well known. I would say almost synonymous with this rhizosphere microbiology. And with the change of concepts and change of technologies, now the microbiome has taken over the conventional microbiology. So quickly, Pius has adapted the technology what people are practicing in microbiomes, and kind of students and faculty who are leaders in the field of microbiomes right now. He is very prolific in his publications, top in journals. All his students are well renowned in their own field, more so in the field of microbiomes. So I really don't want to stand here. Let us listen from him, his perception about microbiome, and how it is going to get into the even the cropping program. Maybe after this lecture, probably we'll listen from our joint director and others. I want a kind of discussion forum and discussion points coming from all of you. So with that brief introduction, I call upon Pius Raimaker to very important talk become using maps to engineer and microbiomes. And I'm seeing some fan readers also. Right? Yesterday when I looked at this word, he was very clearly telling I want the plant physiologist and breeder to witness in the presentation. Thank you, plant breeders sitting here. Let us listen from him and yeah, let's have some discussion. Yeah, yours. Floor is yours.
Okay. Okay, let's first have a sound check. Is it coming over clear? Yeah? Okay. Thank you, Kumar, for this nice introduction, and it's really a great pleasure to be here. My talk today is about engineering plant microbiomes for a more sustainable food production. And the picture I put here, the bubble on the picture, it has many different meanings, a bubble. So if you look at the, at sort of like the image that is projected onto the bubble, it's not very clear yet, right? So it sort of like mimics that in this field of research, we do have some points on the horizon, but how the horizon actually looks like, we are not fully sure where to go. The other meaning of the bubble means that if you now look at funding for microbiome research, it's immense. There's a lot of investment there. And that also comes with a promise that we have to sort of like live up to the expectations of the use of microbiomes for sustainable food production. And sometimes I have the feeling that we're living in a bubble, that if we don't get at some point some really nice success stories, this bubble will burst, and then basically, we're gonna go into a new direction. I have always a positive attitude. I think where we will go is a bright future, but there's many steps to take. And in my talk today, I will present you a few results from work that we published, but most of what I will present has not been published before, so it's work in progress. So, and it's just to give you a feeling of the directions where we are going. Now, before I start with the lecture, I want to make some thanks here. So the first thanks is to this magnificent group. So the last week, it was quite intense, and imagine we are just about halfway. Excellent student group. It really gave me energy. In my new position, I still do some teaching, but not to the level that I used to do. And being here in this intense course, it actually gives me a lot of energy. So thank you to the students for having this interaction and giving the energy. Also thanks to the organizers and also the funding. Yeah. And to the funding agencies to bring me and also my team, so Victor Carrion, Luisa Arias, and Guillermo Guerrero, to here. This was, a beautiful experience for all of us. So I hope you learned from us. Vice versa, we also learned from you here. So thank you for that. I put some Hindi here, I think, and if I copied correctly, it should say thank you very much. Is that correct? Okay, good. You never know what you copy from internet nowadays. So, okay. Then the other thing, also a thank you, is before we came here, there was a team behind the scenes who made this all happen. And this is just a few pictures here. So you see the picture here with, in the lab practicals, but also from this weekend when we went to Jaipur. And again, here, this is not the entire team because several members were missing. Fantastic. So thank you so much. And this is Kumar here. You will not maybe recognize him. But Kumar, thanks so much. We know each other for a long time. Esteemed colleague, but also a very dear friend. So I thank you so much for making this happen with your team and with the students. So, okay. Good, let's go to the talk. My talk has two components. And, but before we get to that, I will first show you a bit an overview of the department that I work in. We have 6 PIs in the department. And the topics that we work on are 2, which is biogeochemistry, which are related to climate change. So greenhouse gas emission, carbon and nitrogen cycling, and we also have a project running in South America, in the Amazon rainforest, where we look at the impact of deforestation on the microbiome composition and how that actually impacts in reforestation. So how can we reforest that area? What is interesting, and I did not know, is that Dr. Ramakrishnan here actually was working in Germany with Paul Baudelaire. He's one of the senior scientists in my department, working on carbon cycling and methane, methane emission. So it's a small world, yeah. And then on micro-plant interaction, which I will be talking about mostly today, we have a group working on plant viruses, evolution and transmission. And Mark Swart is that. That's Mark here. He also works on using bacteriophages to basically manipulate microbiomes, so to cause shifts in the microbiome, but he also works on viruses that are plant pathogenic. Then we work on bacterial and fungal epiphytes. Wietse primarily works on the fungi. And then ECO, which I mentioned here. ECO works on the deforestation and the nitrogen cycle. Then Paulina Garbeva, she does most of the chemistry in my department, working on secondary metabolites, but especially volatile organic compounds and how they, play a role in the communication. And then myself, my own group, we work on microbiome assembly. So that's the last part I will be focusing on mostly. And just one slide about assembly and functions. There are so many similarities, with the human microbiome, but I heard just that there's also an insect microbiome person here. I think, you can also put the insect here. Many of the concepts and principles that we see in plant microbiome research are also in human, insect, any eukaryote that you can think of. And the two major topics in all the microbiome research that's ongoing is how do they impact growth and development, and how do they impact health and disease. And that can be a direct interaction, but you can also have an interaction from a distance, like, for example, our gut microbiota. They also have an impact on mental health, and also in plants you see that microbes on roots can have an impact on what happens above ground, and they call that the root-shoot axis. And there's many groups working on the signaling cascade between roots and shoots, and how that changes chemistry above ground and also resistance to specific diseases. Okay, so usually when I give a talk for a broader audience, plant physiologists, plant breeders, I note, in interacting with groups on that, they are always surprised about, but we work on plant physiology, but microbes also play a role in that. And so I usually have a slide where you can look at the phenotypes of what the impact is of microbes on plant growth and development. And that's the blind date experiment. So a student in my group, he paired 3 microbes with 3 plant species, and then basically looked at the phenotypic readout. Okay, and this is the result of that experiment. I'll walk you through this. So here you have the 3 plant species, a model plant, Arabidopsis. Then we have the medicinal plant, Artemisia, and a food crop. And this is the sterilized control. So this is how these plants grow on these media without microbes. And then what Jae-seung did, he then took out of the freezer 3 random microbes, and he first germinated these seedlings so that they form a root. And then he applied each of the microbes to the root tip, and then he incubated these plants for another 2 weeks. And this is actually the phenotypic readout that you get. So you can see here nothing really happens, or at least seemingly nothing really happens. Here you get some nice, root branching. Here you get primary roots. They shorten, and you get much more lateral root formation. This phenotype, we call that the too much love can kill you phenotype, because, you can see that the plant suffers from this microbe. And then, of course, here, this is actually the ideal world, what we're looking for, that these microbes give a huge boost of biomass, both above and below ground, right? There's no blind date without chemistry. I'm not going to talk about that, but the student also profiled the chemistry in the leaf. So how do these microbes actually change chemistry? And it's amazing. So even with a sort of like a seemingly no response of the plant to this microbe, when you go to the chemistry of the leaves, you do see a change in chemistry when the microbe is applied to the roots. And one of the applications could be that you can, in terms of breeding, or especially microbes, that you could use microbes to boost the levels of high-value natural plant products in these plants. And I know there's a number of groups working on medicinal plants and how you can use microbes to boost the levels of these medicinal properties of these plants without breeding, without genetic modification of the plant. Okay, so in terms of the topics, in terms of health and disease, I will come back to that in the second part of my talk. So once you have identified microbes or microbiomes for a particular plant phenotype, how do you engineer these microbes? So there's a number of ways to do that. You can use a probiotic. I think many people are aware of, ingesting live microbes that fulfill a particular function. You can also use so-called prebiotics. So prebiotics, that's a substrate that selectively enriches or stimulates the growth of those microbes that you want to be, increasing in abundance and performing that specific trait. I think in the medical field, human microbiome field, they are moving towards symbiotics. That's a combination of probiotic and prebiotic. So you apply a microbe that you want to be there, and you provide the substrate that that microbe preferentially utilizes. So you basically give, by that way, you give the microbe a competitive advantage when it has to, establish itself against a microbiome that is already there, right? Which is always. Then on the other end of the scale, we have so-called community transplants. Some examples, of course, are poop transplantations for people that suffer from colon diseases. It's a very effective strategy. And in the field of my institute, we also use soil transplantations to speed up the restoration of natural ecosystems going from an agricultural system into a natural reserve again. And then, of course, there's always a middle of the road. So that's where the field is moving. So people are moving actually in between those two. Complex, transplants, whole microbiomes, single microbes. So they're moving towards consortia. Okay, so several postdocs, former postdocs in my group, and they are listed here, they wrote a number of review and opinion papers on where should we go with this research area. And the summary of that is in this slide. So where we are going, at least in my program, is I think we first need to go from a descriptive to a much more quantitative scenario. So the delta A here, that's in fact the added value that a microbiome has to a particular phenotype. And in that sense, the classical GEM, so genotype times environment times management, ratio, the genotype, you can actually split that into a genotype, so the contribution of the host, but you can also now make that into the contribution of the microbiome. So both elements contributing to the genotype effect in this equation. A former postdoc in my lab, he has developed a model on that, which you can easily download from GitHub. And with your experimental data, you can use this model, and then it will tell you for a given phenotype what the actual or the relative contribution is of the microbiome in that particular phenotype that you see. Then, I'm not going to touch on that, but it is very important. Modular microbiomes. Usually when you work in a plant system or an agricultural system, you're not dealing with one particular problem. Normally, these problems are not, you are faced with pathogens, you are faced with abiotic stress, and the future, I think, what it is to develop a modularity in your microbiome design, where microbes are complementary, not only, by working together, but also in terms of the different stresses that they can, alleviate in this system. Then, which I will talk about, back to the roots. So during plant domestication and breeding, the idea of this project is that we have lost some traits in the plant, but also we have lost some microbes along the way that have a beneficial function for the plants. So can we bring those back? So we started to work in the centers of origin and the wild relatives of our crop species to see, did we lose some traits in the plant, and did we lose some microbes along the way? Basically, plant breeders do the same. They usually go back to wild germplasm to backcross, or bring some traits back into the germplasm, which confers resistance to a particular trait, resistance to a specific disease. And then a very new direction that we are focusing on, it's called precursor-directed activation. I will explain that in the second part of my talk. It is a well-known phenomenon in analytical chemistry. So you can basically redirect the biosynthesis of specific active compounds by providing a different type of precursor to the system. And we are now applying this in microbiome activation. Okay, so let's go to the two elements of my talk. So back to the roots, I talked about that. And then the second part, which is a very practically oriented project. We go into Africa, and that's a project I'm coordinating for the Bill Gates Foundation, where we are, developing microbes for controlling a specific parasitic disease of cereal crops. And I will come back to that in a minute. Again, like I said, work in progress. So many of the things I will tell about, they end somewhere, and it's not really the end. Yeah, so, okay, so back to the roots. This is the map, the world map, and I'm sure, you know, if you go to India, there's probably a lot more, you know, detail there. But it says the centers of food production, so where the crops are grown, and then the centers of origin. And you can see, so the centers of origin, they are listed here, and the centers of food production, so the crops that were domesticated and actually, you know, further improved, they are in a totally different region. So the centers of food production and the centers of origin are disconnected. So is there, along this disconnection, has there been an impact on the microbiome, and have we lost some microbes along the way? And these several slides, several of the students during the course last week, they already had some flavor of that. There are a few new slides in there. And we started this project with common bean, and then it's sort of like, got a bit out of hand, I must say. So we are now working on solanaceous crops, tomato and potato, sorghum, and probably going to expand into, and also in millet here, working with a group in China, working in Africa here. So this is actually the Bill Gates project. And then here we do work on the centers of origin of the solanaceous plants. So what is known so far on the impact of domestication on the association with microbes? There is nice work done, and it's reviewed by Stephanie Porter here and Joel Sachs a couple of years ago, where they summarize the impact of domestication, especially on symbiotic microbes, so mycorrhizal fungi and nitrogen-fixing rhizobia. And the outcome is that in the domesticated plants, there is less mycorrhizal diversity, and also in the plants, there is a reduced responsiveness to mycorrhization. Same on the rhizobium, the nitrogen-fixing side, less diversity, and the plants, they do less sanctioning against rhizobia that are not so effective in nitrogen fixation. Because in these systems, of course, you know, there is nitrogen supplied to the system. But what about all these other microbes that you find on plants? And for that, together with my colleague, Toby Kiers from Amsterdam, last year, we wrote sort of like a research roadmap, perspective paper, how to study the impact of domestication on all these other microbes in the system. And the first step we proposed is to compare the microbiomes of wild and domesticated plant species grown in either the native soil from the center of origin or in the agricultural production soil. Then what happens to these plants, wild and domesticated one, when you put stress on the system, when you provide, you know, an insect pest or a disease or drought stress? How does that impact on the microbiome? And is a microbiome from native soils or agricultural soils more efficient in protecting these plants from these stresses? Then going more onto the plant genetics, and that's where also plant breeding comes in, because where the field is moving is that can we bring in plant breeders to breed for traits in the plant that support specific beneficial microbes, you know, for a particular trait. So we need to know the plant genetic loci. I will give an example there. And then the underlying chemistry. So is there a chemical basis for the differential recruitment of microbes or activation of microbes by wild relatives and domesticated plants? And can you use that again as a marker in breeding? Okay, so this is the area when we talk about tomato. So I'm going to talk about domestication of tomato, where we work. So this is in the south of Ecuador in South America, in the Andes Mountains, where we do field trials, and where we actually sample native soils from that particular area, and where we also sample wild tomatoes grown at that place. And then what we do, so we do both microbiome profiling of plants growing in that habitat, but we also take these soils, bring them to the greenhouse, and do very controlled experiments where we compare centers of origin and centers of production soils. And then we have a whole palette here of germplasm, so wild tomato relatives, Habrochaitis and Peruvianum, and then also a set of modern domesticated tomatoes. And then you basically try to answer the questions that I just said. Do you see differences in the microbiome composition of the wild and the domesticated ones, both in native and agricultural soils? This is one of the first results. Again, work in progress. We see a clear separation between microbiomes from the center of origin and the center of production. And that's a very significant effect. We also looked at rhizosphere, endosphere, so there's also a compartment effect. There was also a genotype effect here, wild versus domesticated, but that genotype effect was actually very small. So the soil actually has a major impact on basically the microbiome that we find on the roots of these tomato plants. Nevertheless, despite this tiny, difference there, if you then zoom in on the microbiomes of modern and wild ones, we do see, a clear difference in diversity, with a higher diversity in the wild tomato lines compared to the modern ones. And on the modern ones, you also get a selective enrichment of specific groups there. And of course, the next question is, and that's where this slide stops, what we did is we started isolating several of these groups that are unique to wild ones or unique to native soils. And we are now doing a functional analysis. Do they contribute to any of the phenotypes that we are interested in regarding growth, development, health, and disease? So that's where we are moving with this line of research. So then if you go to the second part of the research roadmap, we applied a stress. And what the PhD student, Stalin Sarango Flores, what he did, he tested an endemic insect from Ecuador on these two tomato lines, the wild one and the domesticated one. And he then looked for basically the damage that this insect causes when wild and domesticated are growing in a native or in an agricultural soil. Again, the contrast analysis. And this was the result. Interestingly, the domesticated tomato line was resistant to this insect. So unintentionally, I think, through breeding, they brought in resistance genes that also worked against this endemic insect. And this is the agricultural soil, native soil, and we do a natural, a live soil, and then the second part is a sterilized soil. So we eliminate the microbiome. But for the domesticated one, it didn't make a difference. But for the wild tomato, you can see in the live soil, when the microbiome is present, these plants are protected to the same level as the domesticated tomato. But when you eliminate the microbiome, you can see that the damage caused by this insect was much bigger. So this told us that these wild tomatoes, they rely on the microbiome for protection against this insect pest. And then, of course, we do a lot of taxonomic and functional analysis, and of course we are interested in which taxa are differentially abundant in the live versus the sterile soil, because sterilizing a soil, usually when you then start growing the plants, you do get the residual microbiome there, which is always present. You can't keep the system fully sterile. And two candidates came out, Actinoplanus and the Geodermatophilus. And we have now started to isolate these microbes and again, test them back in the system if they can provide protection against insect pest. Okay, moving to the plant genetics. This is much more an elaborate study, and we have published that last year. So for people who are interested in this paper, it's super dense in terms of data. So I will give you sort of like the headlines here. So what we did, and Victor here was also involved in that project together with Ben Oyserman, a postdoc, and again Stalin. What we did is we used a recombinant inbred line from a cross between a wild and a domesticated tomato. And then you have, all the siblings here going to F6 or F8 even. So that was about a 100, and then the two parental lines, and you grow them in the same soils again, and then you profile the microbiome composition on all these, recombinant inbred line tomatoes. And then you get plots like this where the two parental lines, they separate, and then you have this cloud of, recombinant inbred line tomatoes that are scattered here. And then what Ben did together with Victor and some statisticians, we did genome-wide association mapping to identify plant genes, but also genes in the microbiome that associate with each other. because that's a first step towards that. And this was basically the end result. So on a number of traits here, water balance, nutrient cycling, hormones, secondary metabolism, and also exudates and cross-feeding, we find an association, a statistical significance association between, genetics on the plant side and the genetics on the microbiome side. And just to zoom in how this looks like, we get these sort of like metro maps where you have, this is the chromosomes of the plant with the QTLs represented there, and then here we have the microbiome represented. And there are many things that you can extract from this figure. First and foremost is that the association, for example, of this streptomyces, which was predominant on the modern tomatoes, you can see it's not sort of like one region on one of the chromosomes. It actually associates with multiple regions on different chromosomes. So it's not a one gene association. But what was interesting, if you look at the QTL on chromosome 6, we found 84 genes, and we have an iron transporter here. We have an aquaporin, which is related to drought stress and water balance. And on the bacterial side, for example, thinking about the iron transcriptional regulator, we find 2 siderophores, which are compounds that are also involved in iron acquisition in the bacterium. Okay, what does this all mean? Validation, right? So what we did is we started to isolate this Streptomyces ASV5. We sequenced the genomes, and we were able actually to map, to link the genome of the isolate collection that we had with the information that we had from this microbiome analysis on the real population. So we had an exact match there. And then we did the experiment using a transparent soil system where we applied this Streptomyces. And the first question was, does it preferentially colonize the modern tomatoes? That was because that is what we observed in the real experiment. And yes, it did. So that was about a 10-fold higher density on the roots of the modern tomato as compared to the wild. And interestingly, we also saw a growth promotion for both the modern tomato and for the wild one. And for the modern one, it was a much more significant growth promotion. So having this information, we then also, and we're doing the same with the whole real population, we started to profile the exudates that are released from these plants, and can we find metabolites in the root exudates that can explain the differential recruitment of microbes either to the wild or to the modern one. And there's one result here. This is a first PCOA plot. So we extracted, these are the soluble exudates. And interestingly, and to my surprise, the exudates of the modern one, the overall composition, separated from the overall composition of the exudates from the wild one. We are now looking at which metabolites in these exudates actually, are underneath this separation in the PCOA plot. And then, of course, with the microbes that we have, we can see, can you use these substrates to actually show that these are responsible for the differential recruitment of specific microbes. Okay, so with this full picture, what we are trying to do is we're trying to build knowledge on assembly, to use that in engineering plants that preferentially recruit microbes with a beneficial effect. That's where the field is moving at this moment. Okay, good. Let's go into some practical project, which is the project funded by the Bill and Melinda Gates Foundation. It's a project it's the project called PROMISE. And it's a project where we work on parasitic weeds and on sorghum. So this is the crop that we're working on, sorghum. I'm not sure about Kumar here in India. Sorghum is also an important crop, right? Yeah, exactly. So this is a bit of the ideal picture how a field should look like. Sorghum, again, 5th cereal crop worldwide. It's drought tolerant. It's used for multi-purposes. And just the scale here, it's super important in Africa for about 350 million small-scale farmers. So that's the magnitude of the crop. But there is a major problem. This looks like a beautiful picture, but it's not. So, you know, these beautiful flowers here, that's the parasite. So that's the plant parasitizing the crop. And just to show you a little detail here, how it looks like from close, you can imagine that, you know, looking at this, this is so devastating that many times fields are totally destroyed by this parasite. This is just a rough estimation. I was talking to some people and they said, yeah, Jos, you are probably even on the low end of the magnitude of the problem. And this is the distribution of this parasite across sub-Saharan Africa. During my master's, I worked on this one, Orobanki, on faba bean. Also beautiful, beautiful. They are super beautiful, but also devastating for crop production. And I think one of the major problems with these parasites is that they produce so many seeds per plant, which are then deposited in the top layer of the soil matrix, and they just wait until a host comes along, produces the signal that triggers germination, and they can stay there for 5, 10, 15 years, just wait. So the seed bank, you can imagine, if you want to control a disease or a parasite, you go to the life cycle and you target a very sensitive element of the life cycle. And in the Gates project, I will tell you, we actually targeted different stages of the life cycle. But before we went there, I had never seen the seeds myself. So the first thing we did, let's do some electron microscopy, scanning electron microscopy, how these seeds look like. So this is a batch of Striga seeds. You can see these beautiful surfaces. And just to give you an idea, there are about 200 micron in size. So super small. And then when you zoom in on one of the seeds here, you can see that in the valleys of these surface structures, you can see nicely microbes lined up. So we saw fungal propagules, we saw bacteria. Anyway, and then I got excited. So I thought, okay, so this is a system where microbes are prevalent. And the first thing we did, and it also took me by surprise in terms of how, you know, how dramatic is this seed bank actually. And there are many ways to look at the seed bank, but they were mostly physical type methods to separate the seeds from the soil matrix, super laborious. And two years before we started the project, there was the genome sequence of Striga asiatica was released, and we said, let's look for some markers in there, molecular markers, and can we develop a molecular diagnostic tool to quantitatively detect the Striga seed bank in soil. So this is the team back in Ethiopia where we do most of the work so far. And so what they did is, so here is Addis, and they went along the sorghum belt in Ethiopia across an area of about 2,500 kilometers, and they sampled about 50 soils from these regions. So this is a close-up of the areas where we sampled all the soils, and we went all the way up to the border close to Sudan, and this is actually also the center of origin of sorghum, and most likely also of the parasite. And again, this is the team that did all the work. And again, we developed some primers for specific genes in the genome. And then using these primers and molecular diagnostics, we were able to develop a quantitative method so we can actually, you know, tell exactly how many seeds are present in these soils. And we did that for all these 50 soils. And indeed, about 70% of all the sampled field sites were infested with Striga. And in those field sites, we also monitored the Striga incidence, and we tried to infer a relationship between the seed bank and the incidence. There's a lot to say about that, but with the data that we had, we found a sort of like an asymptotic relationship. So with increasing densities of seeds, at some point, you know, basically the incidence levels off, which is normal. Okay, so where do the microbes come in? So again, we go to the life cycle here, so I'll walk you through this. We have the striga seeds deposited in the seed bank, and they usually, like crop seeds, they have to go through a conditioning stage. So it's an imbibition stage that they have to go through. And then when they receive a signal from the plant roots, so the host plant, and those signals have been identified as strigolactones, they germinate. Now, these strigolactones, they are not produced by the host plant to attract the parasite, but they have been, they are released by the host plant to attract arbuscular mycorrhizal fungi, because these signaling compounds, they are regulated by phosphorus and nitrogen. So anyway, but basically the parasite has hijacked this signal, and that's why breeding for resistance is not an easy task, because if you eliminate the strigolactones, you also eliminate the beneficial association with the mycorrhizal fungi. So it's, but there have been really good attempts, because the Gates Foundation, what they did, they have a program on microbiomes and striga control. They have a breeding program on striga resistance, and they have even also a program on chemistry, new chemistry, how to control. So there is a group in Saudi Arabia, and they have developed these analogs of these strigolactones. And if you apply them to the seed bank before the host plant is there, right, so a pre-plant application, those striga seeds, they will germinate, but there's no host. So they have a short lifespan, and that's what they call suicidal germination. So they germinate, and then they cannot find a host, and then it's over. So we're teaming up with this group, microbes and chemistry together. Then later on, there's also some super interesting chemistry here, because after the germination, the germinating seed, they have to form a so-called haustorium, which is a feeding structure, to establish the xyleme-xyleme connection. Here also chemistry, so compounds like DMBQ, vanillic acid, syringic acid, those are haustorium-inducing factors. So the plant releases those, and then the haustorium is induced. Again, those compounds, they are not, to establish a parasite interaction, but they have, again, other functions in the plant. And then you get the attachment, you get the xyleme-xyleme connection, then the plant starts to grow, and then at some point it even starts to photosynthesize itself, But during this stage, you can see it starts to basically drain the host plant, and it keeps the host plant alive until the point that it's up and starts setting seeds and reproduces. So a super elegant system. So with the microbes, we have focused on, can we find microbes that target the seed bank? That was one. Can we microbes that target the haustorium formation? So that's where we focus on with the microbes. And then a third line of research, that's colleagues in Amsterdam and in the United States. They focus on, can we find microbes that trigger an induced resistance response in the plant that basically prevents or suppresses the attachment and the xyleme-xyleme connection. So we are doing a multi-staged effect. I will only talk about results of this stage here, the dormant striga seeds, this very initial stage. If people are interested in this story with microbes and also this story, there is a bioarchive paper from my colleagues from Amsterdam and from UC Davis. That actually, there's a paper now there where they can nicely show that microbes can indeed degrade haustorium-inducing factors and thereby reduce infection. Super nice work. Okay, so targeting the seed bank. Wietse de Boer in my department and Raoul Masteling, a PhD student, they said, if we want to have microbes that work in a complex soil matrix, we cannot depend on a direct interaction of the microbe and the striga seed. We need to have a mechanism in place where microbes produce compounds that diffuse through the soil matrix, to target all these seeds that are there. One striga plant can produce about 20,000 striga seeds, just to give you an impression about the magnitude. And then we came to the work, and we do a lot of work on volatile compounds produced by microbes. And this is the simple assay. So we have a split plate assay with a divider. We grow microbes, bacteria, fungi on one end, and on the other end you have a little disc here which contains the striga seeds. So what we do is we expose these striga seeds for some time to the volatiles produced by the microbes here. Then we take this filtered paper disc with the seeds to a new plate away from the bacteria, and then we apply this strigolactone that triggers the germination. And then you can, and we have a high throughput automated method now where you can screen large numbers of microbes. Do the seeds still germinate? Do they form an haustorium, etc., right? So, and that's what, and there's many things to say, but I'm just going to focus on an example here. Control plates, then bacteria that give a 50% reduction of seed germination, bacteria that give a 95%, and bacteria that don't give any inhibition. And then you can do a differential analysis here, and this group of compounds here, those were sulfurous type volatiles produced by microbes. But we also detected terpenes that are also part of the interaction with the bacteria and the striga seeds. As a proof of concept, we started to look at the sulfurous volatiles. So what we did, you can buy them because they are synthesized here. And then we did a dose response assay with increasing concentrations of DMDS and DMTS. And here is the striga seed germination. And what we see is that with increasing concentrations of the two volatiles, they basically germination is down. DMDS inhibits germination but does not kill the seed, because we also did a viability staining with tetrazolium, but DMTS, it does kill the seed. And the other approach, I'm not going to talk about that, is to make mutants in these DMDS producers in a particular gene, and that comes back later on, is methionine gamma lyase. That's a gene involved in DMDS biosynthesis, okay? So those are the two approaches. And then we were sort of at a crossroad. We screened many microbes for this particular activity, and then especially with these volatile producers microbes, I said, what are we going to do? Are we going to design probiotics? So are we going to design products with microbes that produce these types of volatiles? Again, just to give you the magnitude of the area that we are talking about, it's massive, right? And again, the Gates Foundation, they always work from the perspective it needs to be effective, and it also needs to be affordable for the small-scale farmers. Can we use a prebiotic to stimulate the growth of these volatile producers? That was what, or can we use a synbiotic where we combine both? Or can we use the pure compounds? That's also very tricky. And then Raoul, Wietse, and I, we sat together, and I still remember the time I said, well, maybe we can use another approach. Maybe we can use a precursor. So we're not going to deal with microbes. We're going to use the potential of the microbes that are already present in the system. Because the biosynthesis of DMDS and DMTS is actually quite simple. The precursor is methionine, the amino acid. Then there is a one step with methionine gamma lyase. It goes to methane thiol, and that then oxidizes to DMDS and DMTS. There's one more enzyme here involved. I will come to that in a bit later. So this was the idea. And the first thing that we did is, well, you know, that would be too good to be true. So let's do a simple test first in the lab. So again, the split plate assay here, but now on one end we put soil with and without methionine. We have the seeds as the phenotypic readout, because there you can look at the germination. But we can also trap the volatiles in that same system to see if the volatiles, when you apply the precursor, are they produced, yes or no? To my surprise, you know, it's the control soils, and indeed when we add methionine to the soil, you see DMDS popping up and DMTS. Then we did a dose response and also an exposure time. So with increasing concentrations of methionine, you see that DMDS production goes up, and Striga seed germination goes down. And the exposure time needed to bring it to suppress the germination completely is only 48 hours. So we also did a time series there. Interesting, important here, this is crucial. We also did, this whole story in gamma sterilized soil, so eliminating the microbes, and there we don't see any effect of addition of methionine. So we need a live soil, we need microbes there to give this phenotype. Okay, that's all in the lab, and then we had a two-step process. So again, we had, of course, access to all these soils from these different regions in the sorghum belt. We brought them to the lab. We built a greenhouse in Ethiopia. This is Tayetesima, coordinator in Ethiopia, a good friend of mine. He's the national coordinator of sorghum research. And we did the experiment in the greenhouse, on site, under controlled condition, with 4 field soils. A push-pull field soil, and then 3 soils which were physically, chemically quite different. And then we could see that and this is just an example how the phenotype looks like. So these 4 are with methionine and without methionine. And in all 4 soils, we could get almost a 10-fold reduction in Striga attachments per root dry weight. So that worked very well. One of the questions we had is if you apply methionine to these soils, which microbes actually respond to this precursor? And the whole biosynthesis of these volatiles, is that by all microbes or is it just a specific group? So what Raoul did for one of the soils so far, he did a meta-transcriptome genome analysis, and then he looked at which genes are activated when you add methionine and to which genera do these genes belong to, right? Okay, so, and we did, again, we only need 48 hours to get the phenotype, but we did a time series from 12 to 24 and 48. And already at 12 hours, methionine addition already separates the transcript profiles. So the microbes, they immediately, and that's well known, they immediately respond to the precursor. And then at 48 hours, it's maximum. And then we looked at the genes. And again, these are these plots where, and the top ones were methionine gamma lyase and methionine methanethiol oxidase. And again, here is the chemistry where these enzymes operate in the production of DMDS and DMTS. So that was a beautiful, there are some more genes, expressed here, which are also interesting, but this sort of confirmed that what we are looking at, on the chemistry side, we had a confirmation, but also on the genetic side, we had a confirmation that that's actually true. And then when we map the genes that are expressed to the taxa that belong to that, it was mostly actinobacteria. So Streptomyces and Microbacterium. They were the ones in which the majority here, where the methionine gamma lyase was expressed. And the second enzyme, and we're still puzzled here, methanethiol oxidase, actinobacteria were a minor one, and that function is apparently done by other groups of microbes. So we're still trying to sort out if they work together or... Anyway, that's where we are at the moment, and we are also trying to screen now multiple soils. If for all these soils, it's always the same that respond to the methionine. Okay, and then the real deal. So already last year, we decided let's test this in the field. So we had a randomized complete block design in the field with lots of controls, etc. And in some plots, we applied methionine, and in some, the controls, etc. So this was the field plot, and then intermediate, this was the result. Methionine treated, untreated. And we also, during the course of the sorghum experiment, we also monitored Striga emergence. And even in the picture, the quality of the projector is not so good, but here you could nicely see a lot of Striga plants, and here just a few. So at the final harvest, what happened, this was medium. This was about halfway, a bit beyond halfway. And then there was a severe drought stress period in Ethiopia, which basically destroyed the entire field trial. So when it comes to the final sort of like phenotypes, we don't have data yet. but the field trials are now being repeated again in the next couple of years. So I think this is a nice example what microbes can do for plants if you know which traits to trigger, right? Okay, so I'm finishing up here. So these are the groups that we work with, both in the Netherlands and, of course, Back to the Roots International. A lot of groups here with different types of expertises, evolution, chemistry, plant breeding. So it's a full team of people, molecular microbiology, also Dutch companies we work with a lot. The international team especially, these Back to the Roots programs going back to the centers of origin, it seems simple on paper, but to organize this is super difficult because you got to get access to those regions. You have to have the permits in place to sample and what are you going to do? A Nagoya protocol comes in here. Then with some groups in the US and also in Copenhagen. And I hope with these projects, we're going to make a difference for the small-scale farmers in sub-Saharan Africa. This is a picture I took when we traveled through Ethiopia. This is not sorghum, this is teff, which is one of the local cereal crops. And so with this, I just want to have a quick announcement for those who are interested. There's going to be the Plant Microbiome Symposium in Quito, in Ecuador, center of origin. So if you want to go back to the roots, please join this meeting. And with this, I want to thank you, Kumar, for giving this opportunity. And again, thanks to the team, the students, for having a very memorable 2 weeks, also on behalf of Victor, Luisa, and Guillermo. So thank you, Kumar. Do we need to stand? Yeah, thank you. I think I should stand up now. Yeah. Yeah. No, I'll stand up. You need a microphone?
First of all, hearty congratulations. It's a nice description about microbiomes. I appreciate your efforts, and I have one small clarification. During your talk, it's related to the response of microbes, especially to the precursors like Microbiome and gamma layers, for which you have mentioned actinobacteria and streptomyces, and they have responded very well.
Right.
I'm wondering that how many number of sites, which this study has been done here, because depending upon the diversity of microbes, depending upon the native soil, the composition of microbes vary.
Yeah.
So had it been done in more number of sites, I think the results maybe, will give a better impression on the stability of the data which we are getting, the response of whether it's actinobacteria or some other microbes. And second one is, are we going to correlate this one with the center of origin data so that, to have a better idea about the plant microbiomes for which the objective you have really, proposed. Thank you.
Right. Yeah. Is it? Yeah. Yeah. Very good question. In terms of the responsive microbial genera to the methionine, we don't know yet. That's, of course, in the pipeline. So we have access to the 50 Ethiopian soils, which are physically, chemically quite different. So that will be, for sure, the next step. I don't know what the outcome will be. Maybe also good to mention, yeah, there's a lot to tell here. So sometimes people ask me, okay, nice, this methionine, but is that affordable for the small-scale farmers? It's not. So this is a proof of concept. So one of the things that we are working on is, especially when you want to make this accessible and affordable for local farmers, you got to go to local resources that they have. Yeah, so and one of the propositions that we have in a follow-up project is to use local resources which are rich in methionine. There's quite a number of sources, either from animal or plant origin, that are rich, so that's where we want to go. One other thing I have to mention is that one of the things that, I mean, the outcome would have been also fantastic. Methionine is also a precursor of ethylene biosynthesis. Now, ethylene by itself gives a stimulation, so it triggers suicidal germination. We did not see that here. In fact, when we did the metatranscriptome analysis, we also looked at the pathways involved and the genes involved in ethylene. They were all down. And why then preferentially this precursor goes to DMDS and DMTS, we don't know yet. So that's another sort of more fundamental point. But if it goes the other direction, the outcome will also be fine, right? Yeah, and going to we have soils from the north, which are in the centers of origin here. And we are also planning to do methionine studies on that. So thank you for the question. Yeah.
Great.
Yeah.
Shilpi. Shilpi is from IIT Delhi, representing biochemical.
I don't think I need the mic, do I?
Yeah, it's audible.
Okay.
No, good. Yeah, let's.
Yeah, brilliant talk, yours. In fact, you answered all the questions that I had about the logistics of methionine, because I was wondering how convenient it is to apply it on a larger scale. But that brings me to the other question also about your work on soil transplantation. How do you see that going? I mean, is it really large-scale application friendly, or
Yes.
what do you think?
Well, I think, and we also discussed that with the students during the course, I think soil transplantation, we don't use it enough to mechanistically understand, you know, the microbes and the mechanisms. From a practical point of view, yeah, usually, no, it's difficult. Unless you are able somehow from a soil that is suppressive to a certain, to sort of like multiply or...
Yeah, so the propagation is...
The propagation, exactly, yeah, of that community that contributes to that phenotype, yeah. Yeah, it would be an interesting project.
Any more interactions? You are most welcome.
There's a question over there, come on.
Yeah, go back. Yeah. Well, represent plant breeding and genetics.
Okay,
The right reader.
cool.
Thank you. Thank you for the nice talk. I was wondering, like, maybe you would have, it must be in the plan, possibly.
Sorry?
It must be in your plan already. Possibly, you had looked over the genotype and microbe interaction, and you had taken from a native environment to a controlled environment, possibly in the Netherlands.
Exactly.
Have you thought about the process of doing the same experimentation in the native environment? As you see, because the pure methionine experiment also, for some reasons, which is obvious, the native environment is completely different. The other factors, other than the soil, is completely different.
Yeah.
So how does the interaction happen at that environment?
Yeah.
I don't know whether it is in the plan. That is one. And the other one is like the interaction, the contrast offered by the native microbes and the so-called modern microbes.
Yeah.
Have you tried looking into the response to inputs in the modern day agriculture? Because it will be completely different.
Absolutely. Yeah. Yeah.
So
Okay, good. Yeah, very good question. So actually there's a new PhD student working in Ecuador now, and he's actually doing the experiments that you proposed in the altitude and in the native region We are designing plots there where we grow the wild ones, of course, that occur there, but we also put the domesticated plants in that area, and we're also going to apply biotic stress. So it's in progress. I cannot give the results yet. And then the second question related to inputs. Yes, of course. Yeah, when you talk about domestication, super important, you're not only talking about changes in genetics, because the plants, they were not only, domesticated and improved, but they also are growing in, a heavy fertilized, habitat. We are not looking ourselves at the impact of fertilization on that, but there are a number of groups that do work on the impact of nitrogen phosphorus fertilization on the responsiveness and the microbiome assembly on wild and domesticated plants. We ourselves, we are not looking into that. We want to go for the microbes that are unique in the native soils and then see if they can be brought back.
In addition to fertilizers, it would be interesting to look at the chemicals which are being utilized in...
Sure, pesticides, yeah, absolutely. Yeah, good question. Thank you. Okay.
So, my congratulations for a very good talk.
Thank you.
Just this small thing which I wanted to know, just because you started with intervention of some microbes you showed which help proliferation of roots and all those things.
Yeah.
Well, we do have all these, I would say, microbial interventions and formulations which we use as inoculants. And later on, you shifted to your lecture to that killing of those weeds or the parasites, where some chemicals are. Yes, you said that it is killing the seeds. So, did you really look into? I don't know. That question was already, discussed about the use of methionine, how it can be economical and other things. But I just wanted to know that because basically it is giving you a pathway where some compounds are produced which are killing the seeds.
Right.
So does that affect these beneficial microbes also? Any kind of studies carried out?
Yeah. Yeah, very good question. So indeed, one of the things that in the field trials that I showed you, we have taken soil samples before application and then at different stages of the crop development to look into what you were referring to, non-target effects. Yeah, because some of these volatiles, they have a broad spectrum activity, and they may also have a detrimental effect on other beneficial microbes already present in the system. So that's what we're working on at the moment. I cannot give you the answer. My guess, it always depends on how long is this effect, and is the resident microbiome, is it resilient enough that after the perturbation with this chemical, because again, we need 48 hours to suppress the parasite, is it resilient enough to bounce back to its original state? We do know that the methionine itself is metabolized super fast. So I think around 3 days, and it's sort of like processed. So it is a short-term effect, and we apply the methionine, I forgot to say that, before we put the host plant in the field. So not during the sorghum. So it's a pre-plant strategy. The farmers can apply the compound, then wait 2 weeks, let the microbes do what they need to do, and then you plant your crop.
Really interesting talk. I have one simple question related to the wild domestic tomato and the microbiome interaction.
Right.
Which one is the pest? Have you taken into account the endosymbionts of the sucking pest? Because especially the group of sucking pests, they rely on the endosymbionts for the colonization of the plants or others.
Yeah.
What about the interaction of the insect endosymbionts with the soil micro? Have you taken into account in this study?
We have not. Very good question. We have not. No. We need an entomologist. Yeah.
Dr. Archana. Archana is the microbiologist and principal scientist.
Now everyone. Excellent lecture, Dr. Jarat.
Thank you.
The start was very good, but with bubble.
The end was not good?
The start was so good that it generated a lot of interest in it. My students are doing training, and I know that it is going very wonderful.
Thank you.
My two queries, which only you can clarify. One, the one thing you talked about that because now we have to go from descriptive to quantitative, especially to satisfy our breeders and physiologists, because they generally don't give much importance to microbiology. So in that calculating delta A, so it is you're taking only some of the phenotypic characters, or yield is what type of parameters? Because you said this is the additional advantage in the phenotype. One query. And second query is, because after doing all these metagenome or these analysis, ultimately when we have to go to at functional stage, or so we have to reach to the microbes. So where that story of synthetic microbial communities is coming.
Right.
So how we will go up to that stage? Because even if we are working in these areas, so we have reached to important points that these will be good for making this community. But how we can propose it now for practical purpose, from theoretical to practical.
Yeah. Yeah, very good question. So predictable and quantitative, I think it's super important. So I agree with the plant breeders and the plant physiologists, because with microbial technology, you have to provide, a consistent and effective and a predictable outcome. And yes, there's always a little bandwidth, right? So in that sense, I fully agree with that. Bringing microbial inoculants to an application, right? That is, I think it depends a bit on, basically the problem that you're targeting. If you're targeting, and we talked also about that in the course, a damping off disease, at a very small window of plant development, I think microbial inoculants could be a beautiful solution. When you talk about problems later on in plant development, especially when you go below ground and you have to reach, a pathogen which is, I don't know, at 20, 30, 40 centimeters, I mean, the requirements that you ask from the microbial inoculant is that it will establish itself in the system. Yeah, it will establish it against the background of other microbes that live there. And then it will do its action at the time and the place where it's needed. It's quite demanding, I think. So that's why I think for some of the issues, we are going to use microbes. For some of the issues, I'm super now excited about the whole precursor business, because there you actually use the functional potential of the microbes that are already in your system, right? Yeah, so. Exactly. And I mean, another thing you can think about with the precursor story is if, for example, you have microbially very poor soils to start with, you could think about adding a precursor with the microbe that has that activity to basically kickstart the system. And then if that microbe then dies or does not survive later on, okay, fine enough. If the window is 48 hours is done, then we're good. Yeah. So there is no silver bullet. Let's put it like that. Yeah.
That's a good question, Dr. Archana, for opening up the important issue.
Yeah.
Hello. Dr. Robin Gobel is heading the division of plant pathology here.
All right, good.
Thank you. We have listened. Very nice talk from you. I heard about you for the first time. I am seeing you.
Okay.
So from your lecture, we have seen there are three categories: prebiotic, symbiotic, and probiotics. And what is the ratio of this group of microbes based on the plant types, for example, legumes, cereals, and others, whether one category or they can share each other.
Yeah, one for all, right? That would be ideal. So if you look at microbiome type studies and you, for example, take a soil and you put different plant species in that same soil, you will see there is specificity among those plant species in the microbes that they recruit on the roots, for example, and probably also on the leaves. So what some groups are doing, they are looking at core microbiomes that you find across plant species, or for example, within a given species, across germplasm within that species. I think that's the way forward, and you can look at a taxonomic level, but you can also, and that's where the field is now much more moving, at the functional level. So do you have a core functional microbiome on either different crop species or germplasm within a given species? So that's one thing. So most studies nowadays, they talk about absence, presence, or relative abundance, right? I think the next stage, and that's also studies that are ongoing, you can have the trait. So suppose that you have an interesting trait of a microbe that colonizes multiple plant species. It doesn't mean that that beneficial trait is actually expressed on each of those plant species. So that's another thing to take into account. Yeah. For companies, it would be beautiful if you have, a microbe that can be beneficial in multiple. And yeah, let's talk about that. Yeah. So what they do, and that's also super interesting from a microbiology perspective, they already have a portfolio of microbes in their company, right? So one of the strategies that some companies I know are following is, can we, through experimental evolution, with these microbes that we have, can we evolve them that they are also operational or functional on other plant species? I think that's where it's going. Because for companies to develop every time a new microbial inoculant for another crop, that is super, super expensive in terms of development, in registration. So I think, I suspect that's where things are moving. Yeah.
Yeah, please go for it. Sorry for that. I was wondering, is someone looking into the variability of the striga populations for the response to methionine?
Yeah. So.
The possibility of mutation rates in the targets which you
have. Not yet, but yes. So we have tested 4 different batches of Striga hermontica and 1 of Striga asiatica. And for the methionine, they responded the same. But I don't know if you go into the field, because there's a very high sort of like evolution within the parasite population. That's why it's still there. How that is going to work out. So in terms of mode of action of these volatiles, we have a working hypothesis. So, and it's quite interesting. We are testing that at the moment. So what we think how these sulfurous volatiles work is that they covalently bind to the cysteine residues in the receptor for the strigolactone, basically destroying the receptor for recognition of the strigolactone. That's our hypothesis, and that's what we're working on. And when we were talking with a chemist, he looked at these volatiles. He said, well, Jos, if the mechanism is that, he said, then 2 other compounds, diallyl disulfide and diallyl trisulfide, those are active ingredients from garlic or from onions, right, the allium species. He said, it will also work. So we did the test. And those compounds were even more effective than the DMDS and DMTS. So that sort of like opened up another sort of like approach in a follow-up project, is that we are now also, in terms of local resources, going to try to see if we use allium crops, maybe as an intercrop, or, in that system, can we, in a very simple way, with these types of volatile compounds from plant sources, can we interfere with the seed bank? So yeah, it tells again that having knowledge about the mechanistic underpinning of what you see, it can open up totally new areas. Yeah, thank you for the question.
New opportunity.
Yeah.
Okay. If there are no more questions, I will call upon our joint editor research, Nathan, to offer his perceptions and his comments.
Right.
And his views about the whole microbiome story, how it can be
I think all microbiologists will also know him, but for the information of the others, those who are present here, he is one of the world-renowned scientists, and he is among the top 1% highly cited scientists of the world. So he took almost 2 week time to come to IRI and train our students. So we are really very thankful and very important. Kumar, yes, he knows him for quite long time. He brings him here, and almost for last 2 years he tried, and finally this happened. And this was a wonderful training. Like the students had an opportunity to interact with Professor Roy Makers, all the Friday night at the 17th night or 18th early morning, and early around 3 o'clock I think. Then they were here in the lab in the morning. And changing the soil microbes, changing the soil chemical composition, physical composition, everything changes when you do conservation agriculture or no till agriculture.
Right.
So for that purpose, I think in the entire world, the IRI has released the first variety which is specifically bred for the conservation agriculture.
Wow.
So I
Wow.
think that is also another important opportunity to see whether the HPCS will be 18, which we released for the first time, whether it has any special trait, any specific microbe association, which it has. Because of that, it is performing better under conservation agriculture. So I will not try to summarize or say anything about Professor Rainer's talk because that will be like very childish. So I am not entering into that one. But I want to say this microbiome in all these components that he has presented, it has a huge potential in the agricultural applications. Because as many of you know that in the last year, the government of India has brought a notification on the biostimulant to regulate the plant stimulating chemicals and microbes in a structured manner. So in that, microbes is one of the important components. Although in the classification that was notified in the Gazette, the microbial live microbes are not there in the categories. Only microbial derived chemicals were there. So in the recent meetings, we discussed to include the microbes also in the category so that microbes can be used for the biostimulation, productivity enhancement, and also the management of biotic and abiotic processes. Already several industries came up with such products in our institute. Also, many of you are working, so it will be going to be a very interesting area. And especially it was through speaking and engaging, and he talked about the domestication as well as the going into the Africa. The Striga problem, which is there in Africa, we also have this problem in large area, and we have this problem. And another benefit he was presenting it's a serious problem in Haryana. So I don't know maybe similar kind of comes from. But nobody is working on it. Nobody. Mechanisms ignoring it. There are a lot of opportunities there for the in this area. From this presentation, you understand that you need a multidisciplinary team to solve this issue. So I'm sure this presentation has triggered new interest in most of you, and we will be using this concept in our future research program.
Interesting.
And I also thank all the those who come here for listening to Raman, the project director, what on the center, and plant pathology and microbiology and how and it was from Dr. Anita is from here, the environmental scientist in the environment science. I think we have multidisciplinary team, and then eminent scientists like from plant breeding, Dr. Gopal, and is there from microbiology, how Sangeeta Paul and Dorian and Abraham. Many scientists are here. I don't want to name everybody, but so for all those who come here, it is really very interesting talk and thought-provoking talk. Folks will get the maximum benefit out of it. And I also especially thankful that you have come from IIT Delhi to here for so thank you everyone, and I thank Professor Rajnagar and his team, and the training organizers, Dr. Kumar and Dr. Ramakrishnan. They almost from the manual preparation time, more than 2 months, they are conducting regular online meeting with Rajnagar's team, and they have put a lot of effort to make this happens. And this is an advantage that he was here, so we thought for a broader audience we can. Thank you everyone for attending.
Thank you.
Follow up on
and my colleagues and students. Again, it's a great pleasure for me to do this board of science for this special occasion. And I thank my for doing an excellent job because one of the videos in Microbiomes, and we had a wonderful opportunity to interact with you personally. And I'll say in the last 6-7 days, some amount of information which you have given probably will take about 6 months to teach the students. And those who attended this course, I said they have got another advantage in understanding the concept, and also making progress. Dr. Victor, Dr. William, Dr. Vivian, you've also contributed so much to us and in terms of giving all the basic information regarding use of several tools, strategies, and the map and what flows we have to follow in our analysis. In fact, I'd say I've been using Dr. This slide to show the importance of good microbiome in many of my academic classes, also in presentation. So whenever I begin my talk, I always use this slide, the good, the bad, and the I'm really thankful to you because he has taken our education so seriously. The way he interacted with this, making sure that everything is perfect, all the details which he has given, making presentation, and also the coursework is unbelievable. And I thank Dr. Naren for taking interest in understanding, giving us a full support, and also to appreciate the role from G in V to G, E into that. And also all the faculty members who are shown to come and attend this microbiome concept, because we are slowly moving to appreciate that we are all living in microbiome. Without microorganism, we won't be making any progress. We want to get a simple thing. And to the students, there are several challenges. Maybe the first challenge is to get sufficient money for your ideas to be implemented. But if you make the right, if you identify the right problem, probably you get good money to do research. And also appreciate the concept of microbiome, not only in your research, but also personal life. But I think that will make a very important, significant contribution in making this work more happy. With that, I thank again once again all the team members and especially Dr. Kumar, who took all the pains in enlightening Dr. Joe in terms of his personal friendship and the collaboration with he has. And he has taken so much pains to bring everybody together and make all the arrangements, form the teams, got everyone helping in making this success. Thank you again. Thank you very much.
That we conclude this keynote lecture by Francis. The trainees after lunch you come back to the
Kumar, tea maybe. Going tea in front. Just check if tea is there. Tea. No, it may be there. Oh, again tea.
Okay. Now, thank you all for coming. Yours is going to be here.
Thank you.
Thank you very much.
Thanks for all your.
It's you.
No.
Great. One point of view, thank you very much.